All right. Good morning. So we changed a bit our plans, and we decided that instead of doing each of us a lesson, we're trying a new thing, and we'll split every lesson so that we make it a bit more, even more interactive, or at least. We hope so. So today is going to be a dive into bacterial metabolisms, mostly principles, and quantitative microbial physiology. And the idea is that we try to understand what are the nutrient requirements of bacteria, how cells allocate resources to grow, and finally, how we model allocation of resources, and when resources are allocated, actually what matters is metabolism, and so try to understand, open this big black box that is microbial metabolism. So, but as basically it's going to be a tradition of this course, we start with a question for you. And the question is, let's say that you are in a lab, and you have E. coli, and you want to grow it. What would be the minimal media in which you would grow it? So what would be the nutrients that E. coli would require to grow? And actually, E. coli is a very unpretentious bug, so it doesn't need many things, but still, you would need something. So again, you have 10 minutes to come up with a recipe. Concentrations are not important, of course, but the most important thing is that you try to imagine what nutrients are necessary for E. coli to grow. All clear? All right. 10 minutes. Ma no.
Ma consigliato di fare di uscire. No, io vado a farla di prima.
Shall we? 1 more minute for the recipe? No one.
Sei contento di aver messo quella cosa sul fiocco?
È pensato.
Hai preso la maglietta col fiocco solo per...
Sì, ma non va sta cosa. Ah no, va. Ah già, ho la maschera. Vabbè.
No.
Pronto? Hello. Are we ready? So, are we ready with the recipe? All right. So, let's start writing down some of the elements. Who wants to start? Okay, fair enough. Some carbon. That's it? Something else like the same type of magnesium? Ions in general? Chromium? Ions in general or specific ones? Iron, yes. I'm not sure about chromium. But it's iron. But it's true, it's cytochromes. Chlorine. Sodium. Right? Not bad. Ole. Okay. Okay, maybe I will ask you, in which forms would you give potassium and nitrogen? No, okay. No, it's okay what you have in nucleic acids. How would you provide phosphorus? This is vague. As a phosphate? This is ammonia, mostly. All right. Okay, I think we're there. This is actually the recipe for the M9, which is the minimal medium that we use a lot to grow E. coli in the lab. Let's see if I can pinch it here. Okay. So yes, basically you start with some salts. As you said, you provide phosphorus mostly as phosphate, sodium and chlorine, and then nitrogen as ammonium. And then you need to add some magnesium and calcium, and then a bunch of minerals, and metals. And then finally, you need the source of carbon. I would say that sometimes bugs grow even without carbon or with very little concentration of carbon, but this is a different story. But yeah, so basically then we can also look at what are the, where is the thingy? Percentage of these elements. So you see that carbon is what constitutes the majority of the cell, 50%. Oxygen, nitrogen is actually 13%. And so basically we could principle write the composition of a cell as C2, and then we have, CH2, O.5, and 0.5. So basically this is this is the composition of a cell, if you want to write it like this. And the other question that we can ask is where all these nutrients go. Why do we need so many things? And of course, you can imagine that, actually, so this is a very nice drawing. So here you have all the things that are essential. So basically hydrogen, carbon, nitrogen, oxygen, phosphorus, sulfate, and selenium. And so these are called macronutrients. So nutrients that the cell needs and needs a lot of them. And so basically, you know that, well, we can say that carbon is, well, basically goes pretty much everywhere, from cell wall to all the organic chemistry is based on carbon, basically. And the cell uses a range of completely different compounds that contain carbon to harvest energy. Nitrogen, we said, is 13% of the cell content in dry weight, and it can be supplied, here we see it can be supplied not just as ammonia, but actually as nitrate. So there are a lot of bugs that can use nitrate as a source of nitrogen. Fewer bugs can use nitrites, and even fewer can use directly N2. And these are the nitrogen fixators. Yes, it's English, possibly. Okay, and finally, you can also provide nitrogen in amino acids, and in that case, the cell can use it as both as a source of nitrogen and as a source of carbon. And then, of course, there is phosphorus. Phosphorus is essential for phospholipids that basically are the constituents of the membranes, and also is present in the teichoic acid that is a component of the cell wall that some bacteria have. And then the other one is sulfur that is present in several amino acids, including cysteine and methionine, and also is a constituent of several vitamins like biotin, thiamine, and the lipoic acid. And finally, there is all these other elements. For example, magnesium is important to stabilize DNA and the membrane. And the other one that I want to point out the importance is iron. So iron is quite difficult to get. So if you are in anoxic conditions, so no oxygen, iron is present in the ferrous form, is soluble, it can be uptaken as it is, while if you are with oxygen, you have it in the ferric form, and this is insoluble. It is bind to minerals, and therefore bacteria have developed systems to actually molecules that are released outside the cell to bind iron so that it can be transported inside, and these are known as siderophores. And hopefully we will have time next week to look at how siderophores mediate interspecies interactions. This is what we need for that are important component of the ecology of bacteria. And the other thing that I have to tell you probably is that there are some of you before mentioned growth factors like vitamins, probably someone from here. So, and basically you need them because they are coenzymes. And there are the requirements of vitamins are very different depending on the bacteria, and some of them can directly produce their own cofactors, growth factors. Others depend on other bacteria for them or depend on you to provide them in the medium. And with this, I think that it's you. We need to change this. But this is just to give you an idea of what you need to grow, which is what we were.
There was another. Oh, okay, this is for later. Okay.
No, I can. Yeah. Actually, so in the PowerPoint you can find. So I put basically a table in which you can read all the different nutrients and where they go and why they are important.
Thanks. Okay, so basically what we did yesterday, what we did yesterday was to write how models of how bacteria grow, right? So, and sort of the basic form when there are enough resources is that we see that these bacteria grow exponentially, okay? So what we want to do, as Martina said, is to zoom in this description and see what actually happens inside a cell, okay? So let's have a sort of simple description of all the steps that sort of goes from nutrients into biomass. So let's first have a sort of intermediate look where we look at these steps, and then we're gonna make a simplification, and then we are gonna go back and make like a more detailed description, and then we're gonna simplify. So the idea of these lectures is that we are not gonna start from the more complicated to simplify, or the most simplify and make it more complicated, but to jump in between, okay? So we can imagine that we have a cell, okay? And it's big because we want to look inside, okay? So we have nutrients, okay? And here, as you see, I'm already simplifying, right? So I'm already saying there are nutrients, okay? And then what we want to do is transport these nutrients inside the cell, or sometimes transport the product of some reactions to these nutrients inside the cell. And in general, basically, what we have is that we have what we call metabolism, which is, for now, some black box, okay, that takes what is its outside, performs some reactions, and produce, what does it produce? Produce energy, if you want to say, okay. Produce energy, and then produce the product of biosynthesis, okay? So it produces, for instance, amino acids, and produce other stuff, okay? These other fast stuff could be nucleic acids, phospholipids that are used for the membrane, and all the other things that needs to do to make a cell, okay? Then what happens to the amino acids? They are assembled into proteins, right? So then they attach to tRNAs, but then there are like ribosomes, which reads like, sort of go through the mRNA, and then there are tRNA charged with amino acids, and so it creates this sequence of amino acids that then folds into a protein. This is my folded protein, right? And at the same time, of course, there is DNA, and so this is translation. And then there is DNA, which is transcribed to mRNA, and this is what goes inside, okay? So these are all the processes, and basically, when we write the growth rate, what we are doing is that we are writing the flux of these, right? How do we go from nutrients to proteins that make other stuff and make biomass, okay? This is the idea. Now, what is complicated here in this, in all this scenario is that all these things are, of course, related to each other, right? So they all interact. So the proteins that we make are, for instance, enzymes. That is what actually is needed to catalyze reactions in the metabolism. The proteins that we make are the ribosomes that are needed to translate proteins. The proteins that we make are transcription factors. That is what binds to DNA to transcribe specific regions. So what determines which proteins are we making, okay? But also the cell can sense what is in the environment, okay? And the sensing goes through what enzymes, determines what enzymes we produce through binding to amino acids and so on and so forth, okay? So a cell is a machinery that takes decisions based on what is the environment, on how to allocate their resources. So they decide how much ribosomes should I produce versus how much enzymes, which enzymes should I produce, in what amount. So all the decisions on how to allocate what they get from nutrients. So how to allocate energy that they get and how to allocate mass, if you want, or elements to make different things, okay? Now, what we do, this is the complicated problems. How do we build in all the complexity of these feedbacks a simple description? And the trick that I'm going to show now is that the trick is to forget about the decision, okay? So if you think about if you are in a steady condition, okay, a cell is doing something, right? And the decisions that they made are stationary, don't change over time, okay? If we don't change condition, a cell decided to do something and is doing a cell or a group of cells and is doing that forever. So the scenario and the sort of mathematical models that we are going to make are performed in this scenario, okay? But still they can reveal a lot of things about how cells allocate their resources. Okay, so we have this simple but still a bit complicated picture, or more complicated than what I want to do, and let me simplify it even more, okay? So the simplification is that we have nutrients, and then there is a flow, a flux, and these produce amino acids, okay? And then there is a flux, and these produce biomass, okay? So this simple flux, okay? Of course, we neglected a lot of things. There is no explicit energy. We're just following the flow of mass, okay? And the idea is that we are thinking we are in a condition where a cell is in what is called steady exponential growth. What it means, steady exponential growth, it means that we have actually a population of cells, and all of them, they are growing exponentially. So they grow, they divide, they grow, they divide, they grow, they divide. So if we follow the mass of the colony is growing exponentially over time. And is steady in the sense that the growth rate don't change over time, okay? So we have that the mass, okay? If you think this is the case without ecology in some sense, right? So ecology is when there is an environment and the cell is changing the environment and the environment is changing the cell in response. In this case, we are assuming that there are enough resources that the environment is not changing and is not changed by the growth of bacteria, okay? Very good. So the sort of picture we want to put forward of a cell is that a cell is divided in different components, okay? So it's divided in different components, and these components are allocated to these different functions, okay? So we imagine that there are different components, and a cell, the mass of a cell is the sum of the mass of these components, okay? And I want to start with a model, and then we are going to see other iteration of this model where we make this more complicated. But the simplest possible model, or the simplest interesting possible model, is the case where we have three components, okay? Three sectors, okay? So, and these three sectors are, one is the ribosomal sectors, okay? So these are ribosomes. Then there is a P sectors, which are all the proteins that are related to metabolism. So the R sector is responsible for this flux. Then there is a P sector, which is what is responsible of metabolism. So if you want, this is translation. This is metabolism. And then we have another sector, which is, let's call it Q, which is all the other functions. Okay? So if we think of a cell, we are thinking that we divide the mass of a cell into how much this mass is allocated to different functions. Okay? And we are thinking about these three functions, so these two and all the rest. Okay? And of course, these are allocated, they correspond to some mass. And the sum of these three mass give me the total mass. Okay? Now, the assumption that I want to make are two. So, the first assumption is that we are in steady exponential growth. What does it mean? Specifically, this means that of course, the total size of my colony is growing exponentially. Okay. But the fact that it's steady means that the cell is not changing what it's doing. Okay? So it means that all these three components are changing, they're growing exponentially, but their relative fraction is constant. It's time independent. So if we define, for instance, the fraction of mass, which is devoted to translation, our assumption is that it's time independent. Okay? So this grows exponentially with the same rate. So this is time independent. Okay? So if we look at this cell inside and we look at the fractions, they are constant over time. And the same for the other sectors, of course. M, and then we have phi q. Okay? And so, of course, by definition, we have that phi r plus phi q plus phi p is equal to 1. Okay? So we can imagine that if we look at cell, we make a pie chart of these three sectors. Okay? So this is the first assumption. The other assumption is that this q, which is like all the other functions which are not related to growth, so this is like sort of housekeeping, the amount of mass, if you want, that I allocate to this function is constant, is independent of the condition. So phi q is condition independent. Okay? So it doesn't depend on what happens. Is that clear? Any question? Okay. So, yes, can I erase this? Okay, so we have these three components: the nutrients, the pool of amino acids, and biomass. Okay, and if we look at the flux over this, okay, the rate at which I flow mass over this, it's lambda. Okay, this is the growth rate. So we have the dm dt is equal to lambda m. Now, if we are in steady exponential growth, we have these two fluxes. Okay, well, we can first look at what are these two fluxes. Okay, so what is this flux here? This flux here, actually, let's start from this. So the flux through here, it's what? So the flux of mass in this channel here is equal to how many ribosomes that are translating I have. Okay, so these are the active ribosomes, the ribosomes that are actively translating. Okay, times what is called the elongation rate, so how many amino acids they spit out per time, okay, times the mass of an amino acid, okay, which I can write as K MAA over the mass of ribosomes times the mass of translating ribosomes. Okay? So this is the number of ribosomes, is the mass of ribosomes, or of the ribosomal sector to be more precise, divided by the mass of a unit. That's the number. Okay? And this I can write as, I'll call this parameter KT. So this is KT times, in principle, phi r of the ribosomes that are translating times the total mass. Yes?
Shouldn't it be dB dt?
Oh, yes. Well, sorry. Okay? You can follow. No, this is the derivative. Yes, this is the derivative, right? It's the flow over here. So when I start, if I see a unit of mass here, how it's flowing here, okay? So the mass I create per unit time is the flow on this channel, okay? Now here I have the fraction of translating ribosomes. What it turns out is that the fraction of translating ribosomes is actually that there is always a fraction of ribosomes which are not translating. It's still, this is something that is in some sense still an open problem of what they are doing, these inactive ribosomes, exactly why they are not active, why this is more or less condition independent. But this is like phi minus, let's call it phi min. So these are the ribosomes that are not, there is always a fraction of ribosomes that are not translating. So this is the total fraction of ribosomes, and this is the one that are not translating, okay? So what we get is that the mdt is kt phi r minus phi min times m, okay? On the other end, what is the flux through here, okay? How the mass flow through here. This is dm dt. On this, well, that is how many, how big is the p sector, the sector devoted to metabolism, okay? So it's mp, okay? So how much is allocated through that, times the quality of the nutrients. So how much these nutrients, so the things that I have here, are good to, are easy to digest if you want to produce mass over here, to produce amino acids. And so that's a parameter, Kn, okay? And of course I can write this as Kn phi p times n, okay? So to summarize, what I'm doing is that I'm saying I have an overall flow, which is the growth rate, but then I split this in two parts, in metabolism and translation, okay? The speed of metabolism, it's related to two things. How good are nutrients, how abundant they are, what is their quality, what is their type, okay? And how big is the sector, how many resources I allocate to metabolize, okay? What fraction of resources, internal resources, I allocate to metabolize. So this is nutrient quality. On the other hand, the other step, so translation, it's related to how good I am in translating, how fast I am. So it's, let's say, translation speed times the fraction of resources that I allocate to translation, okay? Yes?
Okay. I'm kind of confused with the, like, you have written the flow in these two steps, so the nutrients to the amino acids and the amino acids to this mass. But when you wrote the derivatives, both of them are like increasing m. For me, it would make sense for the derivative of the first flux first to have to pass through the second one and then go to m.
If you want, you can do this. Yeah, you can do the same, but you realize that these are in balance, right? Another way to say this is that if you look at the flux, so the speed, you have the speed of the total, which is lambda. Okay. If you are, imagine that you have a pipe, right? And you divide the pipe in two parts, and then you look at the flux over the whole pipe, which is lambda, the flux over the first pipe, which is this, and the flux over the second part. These three quantities, so the overall and the two parts, have to be equal if you are in balance, right? So that's another way to see. So you can see this as you are growing, and if you have to grow, everything has to grow at the same speed. So everything is going exponentially at the same speed, or you can see it as flows, so per unit mass. This is equivalent. If you want to convince yourself, you can write an explicit equation for this, okay? And then you ask that everything in equilibrium, and you realize that this has to be is basically always proportional to mass. So you can write it in the same way, okay? So,
sorry.
Yeah.
I don't know if I understood correctly, because like thinking in this path of transforming amino acids into proteins, it wouldn't affect the biomass because
no, but what is confusing is that DMDT is the flow through the total channel.
yeah, and then the other DMDT.
No, these are always the flow through the channel, right? How the mass is increasing over time.
Yeah, but then like the mass wouldn't increase if we're seeing just the parts going from the amino acids to a protein. Like the whole thing are inside the cell. The biomass
This is like to say, I mean, you divide this in two parts, okay? And what you're looking at is how much flow is coming out of here. Imagine really at pipes and flow, okay? So you're looking at the flow here, which is the MDT, right? Now the flow here has to be equal, the MDT is equal to the flow here and is equal to the flow here, right?
Okay, so, but...
So I can measure the flow here, or I can measure the flow here, and if everything is in equilibrium, the flow here, or the flow at the end has to be equal.
Okay.
Okay?
So they are equivalent. But
Yes, I mean, what the MDT is always the thing at the end. Okay?
Because if you, at first, if you do not assume that those two processes are like at equilibrium in the way that they are adjusting itself one to another, the total flux would be governed just by this lower one.
Let's look at another way to do that. Suppose that I follow the mass of the amino acid pool. Okay. So what I can do Let me think a second. Yeah, I can write the mass of the amino acid pool, right? And I say this is like something that is proportional to Kt phi Kn phi n times n, okay? Right? Because this is the MP, so it's what is driving this. Okay, so I say this is the same. Now, since I'm steady state, okay, the mass of the amino acid pool, okay, is a constant times the total mass, right? So it's the same thing. Okay, it's because all these things are a constant fraction of each other. So basically, if you make this thing equivalent, I mean, what matters at the end is you're looking at these things growing, but it's easy to think about fluxes. So you look per unit mass, okay? And these fluxes are the three terms. So what you get is that lambda, okay, has to be equal to that flux over there, which is, actually, let's start from this. Kn phi p, which has to be equal to Kt phi r minus phi min. That's it, okay? So we have this equivalence, okay? So what I can, for instance, write, I can write this equation for, let's say, phi r. I can write this equation for phi r, and I get that phi r here is equal to phi min plus lambda over Kt, okay? And then using what I know about this, the conservation of the sector, I have that this is 1 minus phi q minus phi r by definition. Right? So I can call this as phi max, because this is the amount that you can go for ribosomes. And so I have that phi r is also equal to phi max minus lambda Kn. Okay? Now, these two equations have to hold. They both have to hold. What is the difference? The point is that I have, here you can see that I have these two equations. So you have this equal to lambda and this equal to lambda. So are two equations for phi r and for lambda. Okay? So with this, I can solve for phi r and for lambda. Right? And in general, this value lambda will be a function of, well, these two parameters and both kt and kn and the same phi r. Okay? So before going to that, there is a simple things I can sort of use from this equation to predict the result of the experiments. Okay? So suppose that I change nutrient quality. Okay? I change the type of nutrients I give to the cell. Okay? So if I change nutrient quality, what happens is that lambda, the growth rate, will change, and my resources allocation, so how much I allocate for ribosomes, will change, right? Now, if I change only nutrient quality and the translational capacity, so how good are cells to translate, is constant, Kn is changing, lambda is changing, so this is complicated, but Kt is constant, right? So in that case, I expect that phi r will be equal to phi min plus lambda Kt, okay? Because the nutrient quality is changing, so Kn is changing, lambda is changing, but they are in this relation, okay? So what I expect to see is if I change nutrient quality and I look at phi r as a function of lambda, I expect to see a linear relationship. Okay? So this is something that you can do experiment with. Okay? So you take E. coli, you give different, you have a bioreactor, you give different nutrients, and you see how they grow. Right? So you measure growth rate, and then what you can do is that you want to measure phi r, the fraction of ribosomes. So the trick you use is that most of the RNA in cells is within ribosomes, 85%. So you measure how much RNA you have, you divide it by the proteins you have, and this gives you an estimate of what is these ribosome fractions. So you can have an access to that quantity. Okay? So if you no, what happened? No? Okay. Yes. Okay. So if you do that, so this is an experiment performed in that way. And so what you see on the top line is exactly that experiment, right? So you have E. coli in different conditions. These are the dots. You also have some mutants, and you see exactly the same relation. So this is like RNA to protein ratio, which is an estimate, gives an estimate on the ribosomal fraction as a function of the growth rate. Okay? Then you can do something else. So what you can do is that you say, well, suppose that I'd like to keep the nutrient quality fixed. So fix Kn and change Kt. How do I do that? There are two ways. First, I can give antibiotics that inhibits the translation of ribosomes, or I can use mutants that are different and have different elongation rate of the ribosomes. Okay? So if that is the case, I should use this equation, right? Because Kn is constant. So I get phi r is equal to phi max minus lambda Kn. So what I expect is that if I start with a value of lambda and then I start, for instance, to give antibiotics, or if I use different mutants, if I use different mutants, what I expect is that they have different slopes, because they both follow this line when I change nutrients, but with different slopes. If then I take one cell and I give them antibiotics, I should see that this relation applies. So I should see something like this. Okay? And so this is exactly what you see here. So here you see different mutants, the 3 lines, okay? And you can see that the slope is actually linear with the translation rate. So this K here, which you can measure. So you see that basically they correspond to different lines. They have different values of KT, which is what determines the slope here as you change nutrients. Or you can say, I take cells with different nutrients, and then I give them an antibiotics that inhibits translation. Okay? So what I do is that I start from here, and I go over there. Okay? Like predicted by this. Okay? So very simple model, very easy, gives something that can be predictable, and it quantitatively matches the behavior you see in the data. Okay? Okay. Last thing. Now, if you solve for lambda, okay, you can solve for lambda, so these two have to be equal, so you get a solution for lambda. You get that lambda is equal to Kn, Kt, Kn plus Kt, phi max minus phi min. Okay? So this function here, you see that this function is an increasing function. It's like something which is the maximum, the range at which you can change the ribosomal sector. But then it's something that depends on the combination of nutrient quality and your ability to translate, your translational capacity. So how good is the cell to make proteins, basically. Okay? And you have this combination. Now, suppose that you give to the cell the best possible nutrients. Okay? So you give to the cell the best possible nutrients, Kn will be very large. Okay? If Kn is very large, so Kn large, what you get here is that lambda is approximately Kt phi max minus phi min. Okay? Now, these parameters you know, okay? So you can measure them. So this is more or less 0.55, okay? And this parameter here is 5.2 hours minus 1, okay? So if you convert this, what you get is that this is the growth rate, so you can calculate the duplication time. So the duplication time you get, which is log 2 over the growth rate. So this is the time between two cells divisions. Here, with these numbers that you measure with this experiment, you get 14 minutes, okay? Which is very close to what you see in experiments of 20 minutes, right? So in experiments, the fastest cell can grow, E. coli can grow is duplicating in 20 minutes, okay? So the point is that this tells you what is the limit to growth in E. coli, right? So the limit of growth is determined by how fast you are able to translate proteins, right? So this is that prediction, and what you see in experiment matches the observation you have about the data, okay? So this is the simplest, in some sense, view that you can have about the flux from nutrients to mass, okay? Now what we want to do is to, in some sense, either zoom out or zoom in and see what is in this black box, okay? So this is, in some sense, easier to understand, but what we want to understand is what is here, so how these fluxes work, how do we go from nutrients to the synthesis of amino acids and to the production of energy. So with the help of Martina, we dive into metabolism, okay? The buttons.
Yeah, there's a question.
Yeah. Phi min and Phi max. So you have you remember that I was talking about these sectors, okay? So this is R, and this is Q, and this is P, okay? So Phi max is, how big is R plus P? So Phi max is 1 minus Phi Q. Okay? So suppose that you have all your allocation goes into ribosomes. Okay? So this becomes this is the maximum you can allocate, 1 minus Phi Q. Okay? And this is what happens when you have, when the nutrients are very good. So when Kn is very large, your limits to growth is ribosomes, right? How fast you translate, so you want to allocate everything in ribosomes. So this is the maximum. Phi min is the fact that you are not able to use all the ribosomes that are allocated. There is actually a fraction here, okay, which are ribosomes that are inactive. It's not fully clear why, if are actually ribosomes that are inactive are other things. It's complicated. It's complicated in the sense that it's at the edge. But this difference here, so Phi max minus Phi min, tells you how much you can allocate to ribosomes, okay? To active ribosomes. So it's your translational potential, how much you can translate, how many resources you can put into translation, okay? So this is how much you can possibly allocate to translation, and this is your translational efficiency, how good you are of translating per unit.
The lower bound we found for duplication, how universal is it?
This bound?
How much specificity?
Well, it depends on KT, right?
Uh-huh.
So, I mean, sort of the simplest question I can give is that it depends on KT, and KT is not universal.
So
okay.
when we find these kinds of microorganisms that have very, very long duplication times, is the limiting factor there, or do we expect it to be other effects?
They have a very long duplication. I mean, also E. coli can have a very long duplication time if it is, you give like, depending on the condition, right? So this is sort of saying, suppose that you give the best possible condition.
Or the conditions in which we study E. coli, that is at 37 degrees, which is a very high temperature, and with 20% of glucose. Or maybe 2, I don't remember, 2% glucose, something like this. So they have a lot of nutrients. The temperature is so high that they can actually grow very fast. But if you put E. coli at 4 degrees, it's not going to grow. So it's not just not universal, but it's context dependent, I would say.
Yeah, exactly. So, but this is a bound, right? So there are two parts of the answer, I think. One is that it's a bound, so it's the best you can achieve. And if you take another bug in another condition, you don't know. I mean, that condition is likely not the realized one. Yeah, it's the maximum growth rate. Then the other part, I think, is that this is under the assumption that the limits to growth is determined by translation, okay? Which there could be other factors that limit growth that are not your translational capacity.
Ma una in realtà. Dov'è, qual è?
Con questo qua torni nella lavagna con sopra la slide.
Ok.
Quindi ti muovi diagonale.
Tu questi li fai vedere dopo. Okay, so first thing that we want to make a point about is that why this black box that we call metabolism actually matter for ecology. Because basically now the only thing that we've been talking about is a cell. A cell that we said before is in a condition in which nothing, so it's not the environment is not changing exactly what the cell is doing. But in the end, what really matters, and this thing is not collaborating this morning. choke myself. Okay. So one thing that we can ask is, give me a second. So when you have this cell, and why this black box should matter is that, again, if we have nutrients, how, and not just nutrients, but in, let's say, the energy landscape of the environment, how much the cell can harvest energy depends on the environment, but the metabolism also affects the environment because cells are not a perfect machine, so what is produced usually doesn't remain necessarily inside the cell, but for example goes outside. And this could be protons that change at pH or other nutrients that, again, modify the energy landscape. So this is, let's say, this is the big picture of why we should look into metabolism to understand the ecology of microorganisms. But when we open the box of metabolism, that's what it looks like, which is a huge, glorious mess. I don't know, maybe some of you, can you recognize anything here? The Krebs cycle? Great, because it's a cycle.
Type of macromolecules. The colors are type of macromolecules, maybe?
Not really. Those are pathways. So, okay. For example, this violet here is mostly carbon metabolism. So also the picture is just to the violet goes here. This is the glycolysis, TCA cycle. This is a huge mess that depends on, this is microbes in the environment. But there are a lot of these very nice maps in the KEGG database that is actually is a database that collects and maps all the reactions that have been discovered in microbial cells. But the other important thing that we can say is that this metabolism is generally conserved. So if you take E. coli, or if you take yourself, the reactions are basically the same. And not just that. Reactions are basically the same, but also most of them are of the same type. So yes, you can have isomerizations, hydrolysis, but there is one type of reactions that is universal, and that redox. Redox. Can you make an example of a redox?
Every nutrient reaction that happens in the transfer. Yeah, I would say the
yeah, this is a huge one, indeed.
Oxidation.
Yeah, but also one that everyone knows is actually this one. Right? This is a redox. And we call it redox because basically it's divided into parts. One is so you can write two half reactions here. One is this one. Probably I should turn this. Yes, it worked. I should write smaller. So So this is actually an oxidation, and this is the reduction. And the way we call them, this is basically the most oxidized, and this is an electron donor that is donating electrons, and this is while oxygen is actually an electron acceptor. And you can basically all the majority of the reactions that occur inside the cell and in metabolism are redox. And so basically the water formation is this plus. I don't have space. Okay. Am I saying something completely new, or you remember biochemistry from your studies? Okay. Now tell me if I'm going too slow and I can skip all these things. But basically, we can define substances based on their tendency to donate or acquire electrons. And actually, we can write this, the tendency is the potential that is called reduction potential. And is measured and this indicates that it's measured at standard conditions. So at pH 7, at temperature of 25 degrees, and in relation to hydrogen, H2, which is the reference, and is measured in volt. So by convention, all the reactions potentials that you can imagine are given as half reactions. And in these half reactions, usually they are conventionally written in this way. So you have the this here, and this is basically you write always the most oxidized on the right and the most reduced on the left. And you can get a potential for this particular half reaction that is telling you the tendency to donate or acquire electrons. If this potential is negative, like in the case of this couple, the most oxidized is the one that has the largest tendency to donate electrons, while this one has only a weaker tendency to acquire electrons. In the case of oxygen, is actually the opposite. The potential is positive, and as a consequence, the tendency of oxygen to acquire electrons is always bigger than water to lose electrons. Okay? When we think about now I will change again. No? All right. When we think about reactions inside the cell, what we can imagine is a reduction tower. It's a redox tower. This is telling you all the possible couples. Again, the convention is that you have the most oxidized on the left and the most reduced on the right, and you get all the different potentials. And as I said, all the negative potential. So you see, there is a variety, and you go from actually the redox couple that has the highest potential to lose electron is actually the sulfate, and the best, basically, electron acceptor is oxygen. And when we think about metabolism, you can think about a flow of electrons. And to understand what's going on, you just need to follow where the electron goes, right? So, and you can see that you don't need to go so you can do different jumps. So these couples are not fixed, and the fact that you are an electron donor or an electron acceptor depends on which of the other couple you are reacting, all right? So for example, we can, for example, say our hydrogen can react with fumarate and succinate, and this gives this delta G represents the change in free energy, and is telling you how likely it is that this reaction is going to happen. This is negative. This means that this reaction is favored in this direction. But hydrogen can also interact, react with nitrates, and again, this gives even a larger free energy. But of course, the biggest free energy is when hydrogen reacts with oxygen in the formation of water, and you get a delta G of, we can write it here, kilojoules. So the bigger the jump, the bigger the energy that you can get from the reaction, and as a consequence, in principle for a cell, the bigger the energy that can be stored and used for functions. So tomorrow we will dive more into these different couples, which are telling you how much energy can actually be harvested by a species in the environment, and therefore, basically how fast it can grow and how fast it can compete for others. And this, again, it depends on which metabolism the cell has available, which, again, depend on the type of substrates, electron donors and electron acceptors that you have in the environment. Okay? This is the first organizing principle of metabolism. We can't forget all the different type of reactions. We can just follow the electrons, and we will know basically how good a species can be at harvesting energy. Is this fairly clear? But then you can ask, okay, here you have few couples. So you can, yes, you can combine hydrogen and fumarate and succinate, but this is just a fraction of the reactions that a cell is doing in its metabolism. So what the cells has devised is are basically the intermediary that can match couples that otherwise wouldn't do a redox reaction and favor these reactions. One of these intermediary is the NAD, which is the nicotinamide adenine dinucleotide. No? Oh, great, of course. Okay. But so, and very interesting, this is an intermediary because it doesn't only carry electrons, but actually carries 2 electrons and 2 protons. And you can do a catalytic cycle in which basically, actually, we can look at the redox potential of this couple, and you can see that is negative. So it's minus 32 or something, and therefore, what is the tendency of this molecule? If I tell you, what is NADH doing usually? Great. It's a good electron donor. Perfect. So in the catalytic cycle, you can imagine, I will try to draw it without. Also, how are we doing with time? I can skip it, actually. So I can skip the drawing, otherwise it's going to take a while and we don't have enough time. But basically what happens is that you can couple the reduction and oxidation of the NAD, and in this way you can actually put together 2 molecules that otherwise would not react. And since this is a cycle, basically NADA is not consumed and can be used continuously by the cell. And what? Yeah, no, maybe we can press the button, but in the idea of the drawing, there would be another important thing to remember is that to do this most of the times, this is the substrate, you would need an enzyme. And what the enzyme is doing is basically favoring reactions even when these reactions are exergonic. So for example, if we go back to the water formation, you would say that this reaction is very likely to happen because it has a negative delta G. But what happens is that if you put together in a bottle hydrogen and oxygen, they would not react and form water. Because before these things to happen, you have to break the bonds that there are between oxygen, the 2 molecules of oxygen, and the 2 molecules of hydrogen. And so basically, you have to bring the reagents into an active state. These actives, basically, if you have your free energy, this is G0 first, and you have the delta G of the products. Let's say I call them A plus B. This is, as we said before, an exergonic reaction, so the G of the products is lower, so we can H2O here. But before these reactions to happen, you need to bring the 2 reactants into an active state, and this particular state this particular delta G is called activation energy. So what the enzyme is doing is lowering the activation energy and increasing the rate of the reactions. The way this works is that the enzyme has an active site that binds the substrate, and what it's doing is when the two substrates are aligned, it provides a torque that destabilizes the bonds, and this is what actually is lowering the activation energy. Okay, we said the first principle was the life is redox chemistry. Then you need something that puts together molecules that otherwise wouldn't react. And sometimes you need enzymes, and you will need enzymes that function as catalyzers for reactions. This reaction was exergonic, so not much trouble for an enzyme to work. But sometimes you have reactions that are endoergonic, which means that the delta G is positive and you need energy to break the bonds. So these reactions are coupled usually with the hydrolysis of a compound that stores a lot of energy, like ATP. And possibly I can skip ATP because I see everyone very fond of this. And so I can just say something about the final principle with which we can organize metabolism, that is dividing between catabolic and anabolic reactions, okay? So this is our famous cell. We have substrates, and we have products. And in this catabolic, we are in this part we call catabolism. What happens is that for everything that is reduced, for everything that is oxidized, something gets reduced. So it's a series of redox. And the energy that is produced in the form of ATP can be moved and coupled with the anabolic part that, again, starting from some substrates, then originates amino acids, which are the building blocks for the cell. The only thing is that if you notice, the same nutrients that we use here or that are produced here, sometimes you can produce molecules that can be used as nutrients within the metabolic flow, then correspond to the same starting point from anabolism. So the point that we still don't understand, and this is a huge question mark, is actually how the cells decides to use nutrients and energy, not to break more nutrients, but to make new amino acids. And this is something that it's at the forefront of this type of physiological research, and we still don't know it precisely. So we still have 10 minutes, so you can do. Yes. So the plan for the moment, these were just organizing principles. Next, tomorrow we're going to dive more into what kind of reactions are here and how this catabolic diversity fuels the ecology of microbial communities. And I used the wrong chalk.
Yes, but I need the mic.
Yes.
Yes, so we can start from exactly this point, and actually the boards are perfectly organized. So in the sense that what we can say is this working? Yes. What we can say is that this is what we did before, okay? But now we want to sort of split metabolism in these two parts that are on the left, okay? And so what we say is that to go from nutrients to amino acids, we actually have two parts, right? So what we have is that we have catabolism and anabolism, and so we have these two steps. And so in the middle here we have these substrates. Okay? Now this is something that outputs energy, and this is something that takes energy as an input. Okay? So what we can do is that is exactly the same thing as before. Okay? It's just adding one step. So here we had the pie chart where we had Q, we have R, and we have the sector P. And now what we do is that we split the sector P, which was this, in two parts. Okay? We split it in the catabolic sector and the anabolic sector. Okay? So exactly as before, what we have here is a phi C, phi A, phi R, and phi Q. Okay? And exactly as before, what you can do is you can balance fluxes. Right? We are still in steady exponential growth, so these fluxes have to balance. So this is still the ribosomal part still hold, but I can write the two parts for the anabolic and catabolic sector. So I have here just a Kc, phi C. So this is expressed the split per unit of resource input, and then I have Ka phi A, right? And then again I have, as before, Kt phi R minus phi min. Okay? Exactly the same things as before. Okay? Very good. So what we can do now is again to solve for the equation, right? So all these three things have to be equal, and we know in this case that the condition is that phi C plus phi A plus phi R is equal to phi Q. Plus phi Q, sorry, is equal to 1. Okay? And now, as before, this is equal to 1 minus phi Q, which I can call phi max. Right? 1 minus phi Q is the maximum size of the ribosomal sector. Okay? Nothing changed. So you have this equation, so I can write that phi C is equal to lambda over Kc, phi A is equal to lambda over Ka, phi R is equal to minus phi min is equal to lambda over Kr, and then I have phi Q. So if I sum these terms, right, this plus this plus this, what I have is that I have the sum of these three is 1 minus phi Q, so it's phi max, is equal to phi min plus lambda times 1 over Kc plus 1 over Kr plus 1 over Ka. Okay? This is just algebra. Right? So then I can solve for instance, Kc, 1 over Kc. So I can write that 1 over Kc is equal to what? Is equal to phi max minus phi min over lambda minus 1 over Kr plus 1 over Ka. Okay? And then I can substitute this 1 over Kc over there. Okay? So for instance, I get that phi C is equal to phi max minus phi min over lambda times lambda, so it's this, minus 1 over Kr plus 1 over Ka times lambda. Okay? So in this, it's some phi C max. And of course, this algebra, right? This is just algebra. I can repeat it for all the other sectors, right? And what I get is, what you see is that when I sum these, I extract the Kc, which corresponds to that, and I can repeat for the others. So I get that also phi A is just some phi A max minus 1 over K R plus 1 over Kc over lambda. Okay? The same thing. And then what I can do is that I can do experiments. Okay? So, and I can do experiments in particular in a setting where I work in such a way where the catabolic sector is limited by a carbon source, okay? And the anabolic sector can be limited by a nitrogen source. Okay? So by tuning the carbon source and the nitrogen source, I can, if I change the carbon source, I change the value of Kc, and if I change the nitrogen source, I can change the value of Ka. Okay? Of course, I do experiments, everything works. Okay? Also because I don't do experiments. Oh, sorry, this is skipped. Oh. Ah, was not. What happened? Ah, it's here. Okay. So, what you get? No. What happened?
Maybe you have to present
How do I do this? Just 1 second. We are stuck with a fox. Okay. Okay, great. So what you do is that the same as before. So suppose that you basically here you look at phi c. Okay. So here you look at phi c, and phi c as you change the carbon source, so you change these, okay, and you move in this direction. So these are fixed, okay, and since these are fixed, on the top you move Kc, but the other are fixed, so you get that phi c is some phi c max minus lambda over some times a factor. No. Okay. So this is change C. No, it's still the fox. Okay. So if I change C, if I limit by changing nitrogen, what I have is that I should use this equation. So I should use phi c equal to lambda Kc, because this is constant. Okay? Right? So if I move by changing Kc, I move on this line. If I move by changing carbon source, I move on this line. If I move with the nitrogen, I move on this line. Okay? So this is phi c. And of course the same applied, but inverted with phi a. Right? So you have to look at these. So phi a, you move C, you get this. You move carbon, you get this. You move nitrogen, you get this. So it's the opposite. Okay? So this is the catabolic sector. This is the anabolic sector. Okay? And of course, when you put this together, it's still true that as you change both, right, they both contribute to the ribosomal activity, to phi r. Okay? So in particular, if you write for phi r, you get that phi r is equal to phi r to phi min plus lambda over Kn. Okay? And in particular, what is this Kn? Well, this Kn is actually 1 over Kn is equal to 1 over K C plus 1 over Ka. So it's a combination of the two nutrient quality. Okay? So you get these things where you have a combination. So basically, we have put together these sectors before, and we got everything was working because we got an effective nutrient quality. But then you can sort of split the contribution of the metabolic and anabolic sector. Okay? into this. And so this is, again, another way to sort of get a sense of how much a cell allocate resources in these two parts of metabolisms. Okay? So I think this is the end for today. I think was enough. So we can go and metabolize. And I don't know if, important. So for the discussion on Friday, we started putting things on the folder. So we started putting slides. There is a folder with like discussion 1, and there are the PDFs of the papers. As we said, read them all by division of labor. And we also have a Google Doc with a syllabus where there are references for every lecture. Okay? Good. Thanks. Va bene.
Guys, I have